So I'm Keenan Drever, and this is Jules Hobkirk, and today we'll be talking about CRISPR-Cas9 and Huntington's disease.
Huntington's disease is a rare, adult-onset, autosomal dominant, progressive, and terminal neurodegenerative illness. George Huntington was the first person to provide a comprehensive description of the disease in 1872, and Huntington was the first disease gene mapped to a specific chromosome. Multiple areas of the brain degenerate, with this loss of neurons particularly impacting the neurotransmitters dopamine, glutamate, and gamma-aminobutyric acid, or GABA. The most readily identified symptoms of Huntington's disease relate to the loss of motor controls in the later stages of development. This leads to jerky, involuntary movements that tend to flow between body regions, also known as chorea. In addition to these confronting symptoms, Huntington's disease can also result in a decline in cognitive function, altered personality, and psychiatric symptoms. Cognitive impairments can become evident 15 years prior to motor diagnosis. These psychiatric symptoms include depression, anxiety, aggression, and obsessive-compulsive behaviors, which are frequently treated pharmacologically and commonly result in oversedation and apathy in patients with Huntington's disease. The course of the disease is typically 15 to 20 years, with dementia, mutism, dystonia, and bradykinesia predominating in advanced stages. And the eventual cause of death is typically related to complications associated with immobility, such as skin breakdown, pneumonia, cardiac disease, or infection. However, 25% of patients attempt suicide, which is the cause of death in 8 to 9% of cases. Most Western populations show a prevalence rate of 4 to 8 cases per 100,000 persons. Despite evidence of early onset cognitive decline presented by biological markers, such as brain atrophy, no diagnostic criteria has been established for the consideration of these features. However, despite this inconsistency, studies have shown that Huntington's disease can be detected very early on in the pre-manifest stage through quantitative fluorescent PCR analysis. This PCR analysis can be used to identify the number of repeats in the glutamine expansion sequence as shown on this slide. This non-invasive technique can be used to diagnose Huntington's disease in unaffected fetuses through the identification of fetal DNA in maternal plasma. There are currently no disease-slowing or reversing treatments for Huntington's disease, and few pharmacotherapies have been successful in treating the onset of neurotransmitter degeneration and psychosis. As a result, treatments and therapies have aimed to mitigate the impact of the most severe symptoms, such as chorea and motor control degradation. However, as you can see from the table, the number of neurotransmitters affected by degeneration is significant, so such treatments have severe limitations. Overall, there is not enough evidence available to guide long-term symptomatic treatment of Huntington's disease.
Okay, so the disease is caused by the Huntington gene, which is located on chromosome 14, and this is a PDB file of the protein itself. So it is inherited, autosomal, and dominant, and the disease is primarily caused due to toxic gains of function in the mutated Huntington gene. So essentially, the first exon of the gene has a polytrinucleotide sequence, a CAG sequence, approximately 36. Any more than 36 leads to Huntington's disease, and CAG encodes for a glutamine residue, so the protein itself has a polyglutamine tract in the first exon. Because of the polyglutamine tract, with numbers greater than 36, the protein aggregates in the brain, and this can lead to the complications and symptoms of Huntington's disease. So what this is showing is the first exon here has the CAG sequence that is repeated, which leads to, over there, the mutated Huntington protein because it has more of the CAG repeats and therefore a polyglutamine tract. That protein is what is responsible for Huntington's disease primarily. However, cleavage of the protein or incomplete splicing, that pathway there, can lead to just the first exon with the polyglutamine tract, which can lead to symptoms of Huntington's disease as well. There is also evidence to suggest that the toxic RNA of the mutated Huntington gene can lead to symptoms of Huntington disease alone.
Now that we have a clear idea of the symptoms, prognosis, and molecular causes of Huntington's disease, I think it is time to introduce the potential hero in this story. The type 2 Crispr system for genome editing was first engineered from Streptococcus pyogenes in 2012 by a research team led by Emmanuel Charpentier and Jennifer Doudna. In bacteria, the CRISPR repeat sequences act in concert with CRISPR-associated, or Cas, proteins in order to confer immunity from foreign invading nucleotides. This editing system is a re-engineered version of the naturally occurring viral immunity system in bacteria. The type 2 system is comprised of the Cas9 nuclease and a guide RNA, which directs the Cas9 protein to site-specific double-strand breaks in target DNA. This system was first used in a landmark study of Huntington's disease-induced neurotoxicity in mice. This study, conducted in 2016 by S. Yang et al., specifically targeted the glutamine expansion sequence of the mutated Huntington gene using CRISPR-Cas9-mediated inactivation. Specific guide RNAs were designed to direct Cas9 to the T1 and T3 DNA regions flanking the 140 CAG trinucleotide repeat within exon 1 of the Huntington gene. Cas9 then caused double-strand breaks at these sites, thereby introducing mutation through non-homologous end joining. The T1 and T3 RNA sequences, along with the gene encoding for red fluorescent protein, comprised one viral vector on the top, while a second viral vector contained a Cas9 protein sequence. Both vectors were infected the striatal cells of the mice in the spinal column. The CRISPR-Cas9 mechanism was injected directly into the striatum of HD140Q knock-in mice aged 9 months. At this age, Huntington protein aggregates had clearly occurred. As the Huntington protein is essential for neuronal development, this gene was knocked out at 9 months of age, rather than in the zygote, as is more common in CRISPR-Cas9 studies. Thus, neuronal development was not impaired. However, mutated Huntington protein was detectable in the striatum between 4 and 6 months of age.
So I'll just quickly explain that this type of mouse, the HD stands for Huntington's disease. 140Q, Q is the code for glutamine, so it's indicating that there are 140 glutamines that have been inserted into the Huntington's disease gene, which has then been knocked into the mice. So this study was able to completely deplete the Huntington protein in the striatum of 9-month-old mice in a non-allelic specific manner. And after the CRISPR-Cas9 mutation, the mice had significantly improved motor performance. Interestingly as well, whole genome sequencing of the mice revealed that there were no off-target genetic mutations, which can sometimes happen with CRISPR-Cas9. However, life expectancy of the mice was not greatly improved. So what this figure here is showing, in black we have the wild-type mice. In blue, we have the HD140Q knock-in mice that were not treated with CRISPR-Cas9 mechanism. And in red, we have the HD140Q mice treated with the CRISPR-Cas9 in the striatum. And what we can see is that over the course of 12 weeks, the treated mice drastically improved their motor skill performance. And on the left here is what is called a rotor rod. So it tests how long the mice are able to balance on these turning wheels. And as you can see from the graph, there was significant improvement after the treatment. Similarly for grip strength of the mice. Hopefully further studies into the CRISPR-Cas9 mutation mechanism will lead to a better understanding of what the Huntington gene function is in humans, because at the moment it is not exceptionally clear what the actual purpose of the gene is, just that it is essential for neural development. As a result, it is exceptionally difficult to understand how this method will be applied to human beings and the risks associated with that, because you can't knock out an unknown protein in a non-allelic specific manner if you are unaware of what the function of the protein actually is. Hopefully further studies such as this will lead to a better understanding of the function of the protein and eventual clinical studies into CRISPR-Cas9 treatment of Huntington's disease. Thank you.